Good morning, everyone. I'm Tawfiq, and I'm a PhD candidate at King Abdullah University of Science and Technology in the Kingdom of Saudi Arabia. And today I'll present Beyond Protein Functions, Evaluating Completeness, Coherence, and Consistency of Genome Scale Function Annotations. So the gene ontology provides a standardized vocabulary for protein functions. So it describes protein or any gene product functions, and it's divided into three sub-ontologies: the molecular function, biological process, and cellular component sub-ontologies. So because it's an ontology and its description of protein functions, it enables us to perform large-scale computational analysis. And we usually do this through the gene ontology annotation model, which at a minimum has four components: a gene product, like a protein or mRNA, a gene ontology term that describes the function of the protein, a reference such as a PMID or DOI, and an evidence code to describe the type of evidence that was used for the annotation, which can be experimental based on sequence similarity or manual curation, for example. And this annotation model follows a more reductionist approach. So each annotation is independent of any other annotation to that protein. And Go has tried to overcome this recently using the Go causal activity models, or GoCams, which extend this traditional annotation model to include things like biological context and causal connections between activities or annotations. But there's still some limitations to this because the GoCams are only manually curated, so they're very few. They're mainly human, and they're only focused on pathways, which are only a subset of biological systems. And so the function prediction task, there's many computational methods that have been developed for protein function prediction using many approaches and biological signals like sequence, structure, biological networks, or domains. And they follow the traditional model. So the input is a single protein amino acid sequence, and the output is a set of Go functions that can span the three Go sub-ontologies. And usually these methods are evaluated using CAFA, which is the critical assessment of functional annotation. And it provides a standardized benchmark for this function prediction task, where again, we have an input as a single protein and an output as a set of Go functions that describe the functions of the protein. And the metrics are Fmax, like maximum F score, and SMIM, which is the minimum semantic distance. And these metrics are focused on the individual protein level. But there are other things that we think we need to consider, such as system level constraints and functional dependencies, and the biological plausibility of these annotations. So since we now have better sequencing technologies and better assemblers, we have more whole genomes, and as an extension, also more metagenomes. And so we think it's time to reformulate this function prediction task, where instead of being given a single protein, we can be given a set of proteins or a proteome. And our task would be to describe, predict, and evaluate the functions of all the proteins in that set. And this reformulation allows us to ask new questions, such as whether the set contains all the essential functions for life, whether functional dependencies are satisfied, and whether there are possible combinations of functions within the set. And so we go from individual protein annotation to proteome scale characterization. And so we come up with three metrics for what we call the genome scale evaluation task. And the aim here is to assess the biological plausibility and possibility of the predictions in the set. So the first metric is completeness, where essential functions for life must be present. The second is coherence, where functional dependencies must be satisfied. And the third is consistency, where no mutually exclusive functions can coexist in the set. And so we formalize these metrics as logical constraints based on the gene ontology, inter-ontology mappings, and some manual curation. So for the first metric, completeness, again, it states that every genome must have functions essential for life. So how we came up with these essential functions is based on the M. mycoides syn 1.0 genome, which is a minimal bacterial genome that was created using systematic transposon mutagenesis experiments. So they come up with a list of functions that are essential for life in bacteria. And we manually mapped them to their corresponding Go classes. And most of the time, it was just a one-to-one lexical match. And then we divided these functions into 5 categories. The first is the core category, which contains functions that are universally essential in any bacterial genome. And the others, like glucose metabolism or environmental adaptation, are required for some taxa or some environments, but are not universally required in all bacteria. And so we focus on these core functions, of which we find 22, and they're DNA, RNA, or protein related. They have to do with metabolism or biological process regulation. So formally, we assert that for a set of required functions, a genome is complete if for every required function, there's at least one protein in the genome that is annotated with that function. And here we have the formal assertion, and we also did this for all the other metrics, but I won't go into that today. Our second metric is coherence. So if function A requires function B, both function A and B must be present in the genome. And then there's two levels to coherence. There's protein level coherence, but since Go uses the true path rule, which ensures that protein level annotations are propagated across the Go hierarchy, we assume that this protein level coherence is already satisfied. So we shift our focus to genome level coherence, where if protein 1 has function A, then there must be a protein 2 in the same proteome that has function B. And there are three types of dependencies: process dependencies, pathway dependencies, and protein complex requirements. So to evaluate these process dependencies, processes require specific components. So to evaluate this, we focus on the has-part relations, which are axioms in Go that identify sets of dependent functions. So for example, if we have the term lipopolysaccharide immune receptor activity in the genome, then we must also have lipopolysaccharide binding because they are connected through a has-part relation in Go. The second is metabolic pathways. So pathways require specific reactions. And then we find in Go that pathways are mapped to metapsych pathways, which is a pathway database. And in metapsych, these pathways have enzymes, which are annotated with EC or enzyme commission numbers, which are also mapped to Go. So in the end, we have these pathways and all the enzymes that are required for the pathways, also in Go terms. And of course, in some pathways, there are alternative paths that can complete the pathway, so we also account for that. And third, we have protein complexes, and we specifically focus on heteromeric complexes that require more than one unique protein enabled to form. And to come up with this, we looked at all the children of the term protein-protein complex, and we manually curated them as either heteromeric and requiring two or more unique proteins, or homomeric. And we focused on these heteromeric complexes, and the rule was that they require at least two different proteins within the same genome that are annotated with that term. A few things to note here. So these are necessary, but they're not sufficient and they're not exhaustive. And they also measure our degree of knowledge of how proteins perform their functions. And our third metric, consistency. So some functions cannot coexist in the same genome. And so we have some soft constraints. There are some mutually exclusive functions, such as environmental adaptations like aerobic and anaerobic metabolism. So some bacteria are exclusively aerobic and some are exclusively anaerobic, but there are some that are capable of both. So we call these soft constraints. But we focus on hard constraints and specifically on taxonomic restrictions. So in Go, there are also taxon constraints, such as only in taxon and never in taxon. And for example, if we have a genome that contains the annotation photosynthesis and the annotation viral membrane, and photosynthesis is only in plants, but viral membrane is never in multicellular organisms, there is not a single taxon that can satisfy both of these things, so it would be a taxonomically inconsistent genome. These are, again, also not exhaustive, and they only capture known constraints. So the first question we ask is whether manually annotated organisms are able to satisfy these three metrics. And so here we have a comparison of 6 model organisms such as Pseudomonas, E. coli, and Helicobacter pylori. And when we look at completeness, we find that most of them are complete with the exception of chromosome segregation in Pseudomonas and redox homeostasis in H. pylori. And then we look at coherence, and specifically process, pathway, and complex coherence. So coherence also measures our degree of knowledge, and E. coli performs best across all of the coherence metrics, which makes sense because it's the most studied bacterial model organism. And again, if we're looking at our degree of knowledge, we find that process coherence is higher than pathway and complex coherence across all the organisms, meaning we know much more about processes than we know about pathways or about protein complex formations in bacteria. In terms of taxonomic consistency in these model organisms, out of the 6, only 2 were taxonomically consistent. And for the other 4, the violations fell into one of 3 categories. The first is annotation errors. So in Pseudomonas, we find the term pellicle, which has a constraint that it's never in bacteria. And it's actually a manually curated protein. And when we look at the linked publication, it doesn't make any mention of the term. So we think this was an annotation error. The second is overly restrictive constraints. So in E. coli, we find the term RNA binding transcription regulator, which is only in viruses. But when we look at the linked publication, it does describe experimental evidence of this mechanism existing in E. coli. So we think that while this function usually doesn't occur in bacteria, there are some exceptions and there's experimental evidence of that. So it might be that the taxonomic constraint is overly restrictive in Go. And the third is host pathogen interactions. And we find two cases of this in H. pylori and in tuberculosis, where the terms are restricted to only eukaryotes. And what we find is that these functions actually do occur in the host, not in the bacterial genome. So, for example, H. pylori does negatively regulate T cell proliferation, but it does that in the host rather than in its own genome. The next question we ask is whether computational methods satisfy these metrics. So we evaluate 6 different methods on 1,000 complete bacterial genomes we got from NCBI. The methods were DeepFry, DeepGo Meta, DeepGraphGo, InterProScan, SPRAFGo, and Tail. And for completeness, we find some variable performance. DeepFry had 100% coverage, so it predicted all these essential functions in all the genomes. Tail performed the poorest. For example, the term redox homeostasis was only found in 1.7% of the genomes. And we also find that some essential functions are never predicted by some of the methods. And then we evaluated coherence, again, the performance of these 6 same methods. For process coherence, all the methods performed relatively well, between 75 and 95%, but DeepGraphGo and InterProScan performed best. That was also the case for pathway coherence, where these 2 methods performed best, but all the other methods didn't perform as well. Most of the performance was below 50%. And then for complex coherence, we found that DeepFry actually excelled, and it almost always predicted the protein complexes correctly. So our key insight here is that methods that utilize different biological signals excel at different metrics. So for DeepFry, we used PDBs or protein structures as input, and it was better at predicting complexes. And DeepGraphGo, which is an interaction network-based method, was better at predicting pathways that depend on interactions. For taxonomic consistency, in terms of these methods, InterProScan was the only method that found any consistent genomes, and these were 361 out of 1,000. All the other methods never predicted any taxonomically consistent genomes. And there were a few common violations we noticed, such as viral terms in a bacterial genome, plant-specific terms, and processes that are restricted to multicellular organisms. And so we find overall, when comparing these computational methods, that none of them produce biologically plausible genome scale annotations. So why do they fail? I think an important thing to note here is that none of these methods was developed for this genome scale function prediction task. So there's an optimization target mismatch. They're optimized for protein level accuracy, and they perform very well in that sense when we compare their Fmax. And they're not incentivized specifically for the system level coherence. The second is training data limitations. Most of these methods use a sequence-based split, which creates blind spots. So sometimes there are entire protein families that are missing from the training data. And so there are some functions that they never actually learn. The third is we go back to the reductionist paradigm, where Go specifically is designed for independent annotations. And so the methods also follow this philosophy, and they ignore the system level constraints. So the key takeaways here, first, is that the current evaluation criteria ignore these system level requirements. So we do need new annotation models that express these dependencies and methods that optimize for these genome scale constraints. So in the future, we'd like to develop a formal language for biological function that includes conditional dependencies, quantitative relationships, and takes into account mutual exclusions. And we'd also like to create system-aware prediction methods and extend them to microbial communities and hopefully metagenomes, and to include more types of constraints in our evaluation. And in that way, we can shift from a reductionist to a systems level perspective. So I'd like to thank my advisor, Professor Robert Wendorf, and Dr. Maksat Kulmanov from my group, the Biontology Research Group at KAUST, as well as our funders, the KAUST Office of Sponsored Research, Center for Smart Health, and Center for Generative AI, as well as the resources from the KAUST Supercomputing Laboratory. I also will be graduating soon, so I would like to connect with people here if there are any opportunities. That's how you can contact me. And then that's barcode to our preprint on bioRxiv. Thank you.
Thank you, Ron. That was a good talk. Any questions for our speaker? Paul.
Thanks. That was a really nice and very, very thoughtful, well-reasoned talk. I really enjoyed it. I agree. These are really nice. It's really nice to start going towards the systems perspective. So I had a specific question, actually.
Yeah.
It was interesting to see. Did I understand correctly that you found some taxon constraint violations in the GOA knowledge base?
Yes.
Okay, so this would be really so. I'd love it if you could report these. Do you know about the gene ontology GitHub repository?
Yes.
Yeah, it'd be fantastic if you could actually make tickets to find some of these, because there are QA pipelines that use the taxon constraints.
Yes.
So any violations would be unexpected, and then any taxon constraints that you think are overly restrictive. We'd love to hear about those as well. So
yeah.
Just in general, I just want to plug, since that in general gene ontology, I hope everybody thinks of this as a community project that people can actually help us to improve the quality, and this is really a nice way to start doing that. Thanks.
Yeah, thank you for letting me know.
Thank you. Other questions? Mark?
Thank you. Very nice talk. I was wondering about those taxonomic violations in both automatic and clear. Sometimes viruses integrate into genomes.
Yes.
So wouldn't you expect to find viral functions within the bacterial or eukaryotic genome from the virus integrating in the genome?
Yes, that could be possible. Yes.
So how do you manage this? You call it a taxonomic violation, or do you say it's normal because this piece of DNA actually comes from a virus?
I mean, I think it's a hard distinction to make, but it is something that we should take into account. Yes.
Yep. Have you ever found such? Such things? Or?
So we did. Yes, actually, it was the most common kind of violation when we look at
taxonomic violation.
Yes.
I understand. Right. Thanks.
Very nice talk, thank you. So how are you dealing with multifunctional proteins? I mean, they cannot have mutually exclusive functions, but we know that you may have a protein working translation and on transcription, like the C-term and domain of CRFAH. And how can you contemplate and include this kind of behavior? Because it's not anecdotal in your system.
I'm not sure I understand. You mean if
there are proteins that may have more than one function.
Yes.
So, and they may appear to be exclusive, mutually exclusive. So this is something that you are thinking about integrating in your system.
Yes. So actually, all the proteins that we have, or most of them, have more than one function. We look at the set of functions that are annotated to that protein. But in terms of process coherency, we're looking at the genome scale rather than just the protein itself. But yeah.
Okay, let's thank our speaker once again. Thank you.